Hello everyone and welcome to the Pencil Tutors channel. My name is Nandika and today I will be giving a biology lesson on the tissue organization in plants. Plant tissues can be divided into two different classifications, which is the meristem tissues and the permanent tissues. Meristematic tissues are tissues that are constantly dividing and undergoing the process of mitosis. Permanent tissues, on the other hand, are tissues that are mature and have already undergone the process of differentiation or are undergoing the process of differentiation. For today's video, I will be talking about permanent tissues, but do not worry as I will be talking more about meristematic tissues in next week's video, so make sure to stay tuned for that. Now, for permanent tissues, we can classify them into dermal tissues, ground tissues and vascular tissues. First, let's take a look at the dermal tissues. An example of dermal tissues are the epidermal tissues. The epidermal tissues are the outermost layer of stems, leaves and roots of young plants. This epidermal tissue normally has a waterproof and waxy layer at the upper part which is exposed to air. This layer will help to reduce water loss through a process of transpiration and also provides protection against pathogens and mechanical injuries. These epidermal tissues also consist of modified epidermal cells. For example, here we have the guard cell. As you can see here, the structure of the guard cell consists of two guard cells and an opening in the middle which is called the stomata. The function of guard cells is to control the opening and closing of stomata. This can also help to reduce excessive water loss from the plant. Other than that, we have the root hair cell. The root hair cells increase the surface area of the root for water and mineral salts absorption. Next, we have ground tissues. There are three different types of ground tissues in plants, which are the parenchyma, the sclerenchyma, and the collenchyma. First of all, let's take a closer look at the parenchyma. The parenchyma is the simplest living cell and does not undergo differentiation. It has the tiniest cell wall in between all three of the ground tissues. The cells of a parenchyma tissue are always turgid. There are two reasons for this, which is to provide support to the plant and to maintain the shape of herbaceous plants. Herbaceous plants are non-woody plants. Parenchyma cells are involved in a few processes. First of all, we have photosynthesis. In photosynthesis, the parenchyma tissues are involved in the storage of starch and sugar as well as gaseous exchange. An easy way to remember this is parenchyma. Paren sounds like the word parent. And what do parents do? Parents provide food for us. Therefore, parenchyma tissues store starch and sugar for the plant. Another process it is involved in is the repair and regeneration of plant tissues and vascular systems. Next, we have the collenchyma tissue. This tissue is made up of living cells which mature into cells that are flexible. The cell wall of collenchyma tissues are made up of pectin and hemicellulose. They are thicker than parenchyma cell walls, but it is not the thickest cell wall of all the ground tissues. The function of collenchyma tissues is to provide mechanical support and elasticity to the plant. Lastly, we have the sclerenchyma tissue. Unlike the two previous ground tissues, the sclerenchyma tissue consists of dead cells when they are matured. And sclerenchyma tissues also have the thickest cell wall between all three ground tissues. The function of sclerenchyma is to provide support and mechanical strength to some parts of the mature plants. It also helps in the transportation of water and minerals. Now that we have already learned about dermal tissues and ground tissues, it is time to look at the final type of permanent tissues, which are the vascular tissues. There are two types of vascular tissues in plants, which is the xylem and the phloem. The main difference between these two vascular tissues are the function. The xylem functions to transport water and mineral salts from the roots to the rest of the plant, whereas the phloem functions to transport sugar produced during the photosynthesis in leaves to storage organs such as roots, fruits, and tubers. As for the difference in structure, the xylem is made up of dead cells and the cell wall is thickened with a layer of lignin. The phloem vessel, on the other hand, is made up of companion cells and it has sieve tubes. This long segmented part is called the sieve tube, whereas the blue cells beside it is called the companion cells. As you can see in the diagram, the xylem vessel only has a one-way flow and there is no end wall in between the cells, unlike phloem vessels which has a two-way flow and cell walls with end walls that have perforations. These perforations are so that substances can move through the sieve tubes. Xylem vessels are normally elongated and hollow inside as the cytoplasm is not present. Phloem vessels, on the other hand, comprise of living cells such as the sieve tube and has cytoplasm inside of it. The sieve tubes do not have any organelles such as nucleus and ribosomes as they decompose at a maturity stage. Now that we know the difference between xylem and phloem, let's look at a simple diagram to show the cross-section of these vascular tissue across the plant. First of all, we have the roots. In the roots, we can clearly see that the xylem has a bigger surface area compared to the phloem inside its cells. This is because the xylem is needed to absorb the mineral salts and water from the soil surrounding it. Next is the cross-section of the stem of a plant. In a cross-section of a stem, the xylem is in the inner surface, whereas the phloem can be found on the outer surface of the vascular bundle. Lastly is the cross-section of a leaf. In the cross-section of a leaf, the phloem is normally found at the bottom, whereas the xylem is normally found at the top. An easy way to remember this is that phloem transports food, whereas xylem transports water. Food is heavier than water, therefore phloem is at the bottom and the xylem is at the top of the cross-section of a leaf. And there we have it, we have reached the end of our lesson. But before we go, let's answer a few questions to make sure our understanding on this topic is complete. Now here's a fresh question right off the bat. The diagram shows a cross-section of a plant root. And they ask for us, what is the structure Y? As we can see here, the larger area comprises of this T shape in the middle, whereas there is a smaller area at the sides, which is Y. As we have learned just now, the larger area will be the xylem, whereas the smaller area will be the phloem. So the answer for this question is clearly phloem. Next, let's look at a similar question which they have altered a little bit. Diagram 19 shows the cross-section of a root. Again, which structure A, B, C or D transports sucrose? Sucrose is a sugar and sugar is transported by the phloem. Here, similar to just now, the structure of the phloem is the area that is smaller, not the T in the middle. The T in the middle is the xylem. Therefore, the answer is C. Next question. The diagram shows the longitudinal section of a phloem, which transports synthesized organic substances in plants. The synthesized organic substances in plants are sugar. To be more specific, is sucrose. What is the function of the companion cell? What is the function of the companion cell? Since this question is only 1 mark, we have to go straight to the point. The function of the companion cell is to provide the sieve tube with energy, ATP, proteins or nutrients for transporting organic substances. Next, diagram 1.3 shows the structure of stems which its outer ring is removed after 1 year. Based on the diagram, explain the condition shown. If we recall the cross-section of a plant, the phloem is on the outer layer, whereas the xylem is on the inner layer. Now that we have cut out the outer part of the stem, we have removed the phloem and now there is only xylem left. So, what are the effects on the plant when we have cut out the phloem? First of all, we need to write what we see in the picture, which is the area above the ring is swollen. This is because of the accumulation of organic substances such as sugars and sucrose. This is because it cannot be transported to the area below the ring because the phloem tissue has been removed. The area below the ring will die or will stop growing. This is because it does not receive any sugar or organic substances. And there we go. That is your answer. That's all for today. Thanks for watching the video and be sure to like this video and subscribe to our channel, and don't forget we post videos every Monday, Wednesday and Friday covering physics, mathematics and biology.